We're very pleased for this session to have Dr. Nezami. Dr. Nezami has been a member of our group for a year, and he's doing some really wonderful work out in Fresno, California, and he's here today to talk about genetics and epigenetics. Please give a warm welcome to Dr. Nezami.
Thank you, thank you. I think everybody should give that to you because you are the pioneers in treating cancer, and I'm just a member of the whole group. That said, I really enjoy talking here because it gives me a different feeling compared to giving my presentations at the tumor board locally in the hospital. So everybody's smiling at me instead of looking at me as a criminal. So this lecture is focusing on cancer and epigenetic plasticity or genetic and epigenetic modifications. I'm sure many of you have heard about this terminology. But I need to go over the basics because most of what I do in my practice is changing the gene expression by modifying the epigenetics. And unless you know exactly what the definitions are, it would be difficult to comprehend the whole concept. It's a new concept, even in the world of conventional medicine. It's a very cutting-edge, new, novel thoughts about how the cancer is actually started from beginning, because epigenetics nowadays is blamed even in carcinogenesis. And then we're going to talk about the details of specific modifications later. For the ones that don't know me, I'm a graduate of a USC and UCSF residencies. I completed a fellowship in integrative cancer therapies. 16 years in practice. I'm a medical director of Pacific Medical Center of Hope, some other research corporations. I'm an advisor on board of directors of California Citizens for Health Freedom. I'm an investigator for Best Case Series for National Cancer Institute. And I've been recently consulting with four other physicians and even oncologists for difficult cases. Research principal investigator at National Institute of Health. I have published in many journals, including Integrative Medicine and Clinicians Journal. And as I said, I present very routinely my cases in the local oncology practices. My interests are at treating cancer through immune therapies, genetic and epigenetic modifications, metabolic treatments of cancer, and then managing the stroma. These are the ways that I treat the cancer. And I think what we are now interested in probably has never been explored in the world of conventional medicine. They're not interested for sure in the immune therapies. You rarely see, even if they're aware or familiar with the word natural killer cell activity. Sometimes when I present my cases, the oncologists say, what is natural killer cell activity? And then I have to show them the studies that Dana-Farber has done in Harvard and other studies on natural killer cell activity and how you can actually treat cancer by activating those. Genetic and epigenetic modification will be the talk of the subject of the today. Metabolic treatments of cancer. Many of you are familiar or might be familiar with the glycolytic therapies, and I know that Mark Rosenberg is going to talk about metabolic treatments, and I used to use metabolic treatments more often than I do these days, and I still have, as part of my invented protocol of MTET or modified targeted epigenetic therapy, I use glycolytic inhibitors. And then managing the stroma. As you know, there is a communication between the cancer and the body, and again, conventional oncologist doesn't want to hear or know about what the rest of the body is doing in regards to the cancer. Some of you may have heard the word reverse Warburg effect, and that has to do with the effect of the stroma and its communication with the cancer. This is conventional oncology in my standpoint. They live in a box. They not only think in a box, but they also prescribe in a box. So everything is about the box, 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 box. And well, even in the world of oncology, conventionally, I speak, if you talk about new cutting-edge treatments in the world of conventional oncology, you still see that there are resistance. So talking about chemosensitivity, well, that's conventional. Well, there is resistance. Talk about genetic biomarker profiling. That's conventional, but still you see resistance. So that's why not only we are facing the resistance in the conventional world, but also some of the oncologists that are more younger and they're more educated about their field, they also see resistance. And so that's very shameful, I think, in my opinion. And thinking beyond the box in conventional oncology, I just told you, so some of the examples would be the Gleevec that initially was a tyrosine kinase inhibitor, but now you know that it also blocks the PDGF, and it's also a glycolytic therapy. And I didn't know that until I actually came across a study that was published recently using Gleevec as a glycolytic example of glycolytic therapy. And then, we investigated more to see how it works and how you can actually increase the Gleevec effect and lower the Gleevec resistance. As you know, most of the patients who are on Gleevec, they respond initially very well, and then after a while they become Gleevec resistant, and then you have to go to the second line, which will be Sutent. And you can do things, I shouldn't say conventionally or unconventionally, this is a gray zone. You can do things to increase the sensitivity of the cancer to Gleevec in the long run. Another example is, Herceptin that is used for breast cancer that is HER2 neu positive, but then you see that it's also used for ovarian cancer because they may share the same genetics. And that's why when a patient comes to me and they say, okay, have you ever treated carcinosarcoma, or have you ever treated this type of myeloma, and have you I tell them that, the location of the cancer is not really that much important as the genetics. So you may have a lung cancer that has the same genetics as ovarian cancer, or you may have a breast cancer that has the same genetics as the bone cancer. So the location of the cancer is not as important to us versus the conventional oncology care about that too much. The genetics might be shared. So these are the examples. For example, Zelboraf that has come out for a mutated BRAF, or methotrexate that can be used to hypomethylate the RAS. These are examples of how conventional oncology can even be looking outside the box and exploring the cutting edge, more advanced treatments. My objective and outlines was to clarify some misconceptions about integrative oncology. I think integrative oncology is and must be the way to treat every cancer patient. It's not optional. I don't think it's an option and should not be given as an option to an oncologist to treat a patient just by what is called the standard of care. I think it must be combined with other integrative therapies. Understanding the basics and core concepts of epigenetic therapies, we're going to talk about that in details if time permits. Reviewing some customized targeted epigenetics and effects on chemotherapy. And my protocol, as much as I'm allowed, I will share with you, and then some case studies. So epigenetic plasticity versus differentiation. So what is epigenetic plasticity? Regardless of talking in a cancer seminar, epigenetic plasticity is a terminology that is used nowadays by physiologists, the people who are the pioneers in the field of epigenetics, just to explain how the genes are controlled in their expression in a normal human being. We're not talking about a cancer patient. And how the stem cells basically have the plasticity to become different kind of cells. So we're going to talk about that. There's a very interesting lecture coming up next month in MIT that I will participate, and we'll be sharing thoughts about epigenetic plasticity. There's also another conference coming up this year in France. These are all new ways of looking at the genes and epigenetic coding. In other words, we're not only talking about the genetic codings, but we're also targeting the epigenetic codings, because there is a coding for epigenetics, and if you can explore that, then you can pretty much control what the genes will do in a normal person, and of course cancer patient will be treated that way as well. History and evolution of the definition. The term that was invented in 1940s is well-established phenomenon that plays a major role in diversity of biological processes such as embryonic development, cancer biology, and immune response, among many others. We are recognizing that these are epigenetic inheritance systems through which non-DNA variations can be transmitted in cell and organismal lineage broadens the concept of heredity and challenges the widely accepted gene-centered neo-Darwinian version of Darwinism. So when you're talking about, okay, what you as non-conventional practitioners, as alternative physicians, as physicians that look outside the box, we know that what we have, what we do, what we act, what is combination of environment and genes, right? But this is just the whole concept. The whole picture is that when you have this part of your system controlled by environment and this part is by the genes, when you interact the genes and the environment, then you have the disease or basically your presentation of those genes. But to explore that, this is basically outside the genes. So when we're talking about epigenetics, we're talking about outside the genes, outside the DNA that controls the DNA and is inherited. So it's not in the DNA, but it's inherited, and it can pass along to the next generation through non-DNA component of the epigenes. And so we're going to talk about this in detail. Epigenetic inheritance describes the ability of different states which may have different phenotypic consequences to be inherited without any change in the sequence of DNA. So now you're talking about two exactly similar DNA components that have different phenotypes, different expressions of the genes. And this is, I think, a really nice slide describing it. So you see the caterpillars and the butterflies are genetically identical and have their genes on or off depending the concept of genomics is gene expression. So when the gene expresses differently, the phenotype completely differs. Epigenetics can be defined as a study of genome function that is contained outside the DNA itself and by means of which stable alterations in gene expression are set. How double-stranded DNA is packaged into the dynamic structure of chromatin is crucial for the process of transcriptional control by regulating transcription factor accessibility to DNA regulatory sequences. Chromatin is constituted of nucleosomes which are comprised of 146 base pairs of DNA wrapped around a core of 2 copies each of H2A, B3, and H4 histone proteins. These proteins suffer post-translational modifications that play a prominent role in gene expression regulation and signal transduction pathways such as methylation, acetylation, ubiquitination, phosphorylation, and simulation, which determine chromatin architecture and ultimately gene transcription. So what your mother ate can determine your lifetime outcome through epigenetic mechanisms. So in other words, just like I said, those DNA structures that are the DNA around the histone molecules can express themselves differently, and when it happens, first, it's reversible. Second, it's inheritable. So what you eat now can affect your next generation having cancer or not having cancer with the same genes. We're not talking about mutation of the genes. We're not talking about mutation of DNA. As you know, the old concept was that for you to get a cancer, you have to have a mutation of the DNA. We're not talking about that. We're talking about accumulative, chronic changes in the behavior of the gene caused by environmental situation can be inherited and can cause cancer in the next generation. So we're talking about carcinogenesis in the next generation that is caused by the parents. So you see an epidemic of cancer these days, our parents have done something wrong. Either it's in the environment or they're being exposed unintentionally to something that caused that. Example, you see the yellow mouse and the black mouse. The black mouse has had the maternal supplements with these supplementations that most of them are methylators, increasing the methylation of this epigenetic component, making the other mouse with the same exact genetics smaller, more fit, more healthy. Versus the other one with the same DNA and same genes without any mutation is not diabetic, is not having cancer, is not having all these chronic conditions. So carcinogenesis and metastasis. I can talk about this for at least 20 hours, and I would be sorry if you come up with questions that I cannot go to the details because it's very, very complicated. I'm just giving you some concept of the whole picture. Metastases, I can show you how much the epigenetics are involved in metastases of cancer. And then cancer prevention, when we're talking about prevention of cancer, how you can prevent that. Cancer progression and survival, cancer response to the treatment. So when we're talking about response to the treatment, it becomes extremely important because I'll go over a case that I just recently saw actually 2 weeks ago. She came from Bay Area. She has BRCA positive, recurrent, resistant ovarian cancer, status post-platinum and Taxol treatment that is BRCA positive, and nobody had a clue how to treat her, and I came up with the action plan, sent it to the same oncologist who wrote me back a few months ago that I don't want you to touch my patient. And he looks at it and, well, of course I didn't listen to him because that's my patient, right? But then he looks at my action plan to this resistant case and he sends me a Bravo email that I had never seen such a protocol. We will definitely follow your recommendations. And that's the Stanford and Palo Alto oncologists. So if you look at the cancer response to the treatment, that's the pitfall. That's the problem with the oncology in general, is that it's not customized, as you know. They don't want to know about the genetics. They don't know about the epigenetics if they want to. They're not taught in the medical school. They're not taught in the residency. And so you have to educate them. It is your responsibility as a non-conventional oncologist or cancer care physician to educate and train them. So you need to know the literature, you need to know the science, and you need to sit down and spend time, do the research, talk to them, and convince them, because this, at the end of the day, you want to have a patient who responds to your treatment. That's the goal, right? Okay, so we talked about environment that has principal role of causing sporadic cancer, New England Journal of Medicine. How many of oncologists are trained in the environmental medicine? Probably 0. 30% of cancers are preventable just by diet. How does diet work? Through epigenetics. I just showed you the pictures in animals, right? Changes in prostate gene expression in men undergoing an intensive nutritional lifestyle intervention. These are all basically conventional journals. Folic acid supplementation inhibits colorectal adenoma. Abnormal glutathione S-transferase gene causing decreased enzyme activity resulted in a 4-fold increased risk of breast cancer. So now we're talking about prognostic value in AML. So we're talking about the studies that mostly have been done outside the United States, unfortunately, because the government of the United States probably is not interested in non-conventional treatments of cancer. That's why when I do research, 90% of the research has been done outside the United States, which is a shame. But if you look at the literature, you see that the SNP changes in the genes can be prognostic, can determine the treatment response. So genetics is important because otherwise you don't know which patient is going to respond to which treatment, right? There is actually a study showing that even looking at the glutathione S-transferase can be as prognostic as biopsy. So you have a patient who has a high PSA and the biopsy is negative. And then you look at the glutathione S-transferase, the data shows that if you see polymorphism of this SNP, that is equivalent to having a positive biopsy. So now we're talking about genetics playing such a big prognostic role. Example of the BRCA, as you know, this is the epigenetic changes that causes the BRCA to be malfunction. As you know, BRCA is a gene that is responsible to repair P10, PTEN, and the P10 is an oncosuppressor gene. So if you have a methylated BRCA that cannot repair the P10, then you have increased risk of breast and ovarian cancer. But you have it more often these days than you used to in 1940s. Why? Because you have higher epigenetic negative effects. And that's why you see that people who have BRCA positive nowadays have 67% risk of having the cancer. Genetic interactions with epigenetics. Epigenetics by itself is a carcinogenic step. So now we're talking about prevention. The split of epigenetics and genetics as separate types of defects in cancer is very artificial. And I go to that in a minute because even the epigenetics have genes controlling them. So we're going to talk about that. In fact, according to the definition of epigenetics as genetic information not contained in the DNA sequence itself, current evidence demonstrates that primary genetic defects, mutation in genes with no known primarily methylating or chromatin modifying activities such as growth factor receptors, adhesion molecules, etc., or mutations in genes that in themselves affect DNA methylation or chromatin such as DNA methyltransferases or histoacetylases, are those leading to altered DNA methylation and chromatin changes, genetic control over epigenetics. All classical genetic alterations, for instance, mutations in oncogenes or tumor suppressor genes of malignant cells, eventually affect gene transcription. So you're going to have a transcripted RAS or HER2 amplification, or are transcription factors in themselves, such as p53 and c-MYC. It is therefore not surprising that the machinery of transcription control could be directly involved in the carcinogenesis process. New era in the genetics. I don't think I have enough time to go over all the slides, but this is what I was talking about, the epigenetic codes. And then we're going to talk about the stem cell, because the question came up earlier. I think it was Dr. Kotsanis who was asking Dr. Forsythe, and I didn't hear a good answer. The stem cells are treatable. They're not only treatable, but they can be differentiated. So when a patient comes and asks me about my expertise in regards to the stem cell, I explain to them that, most of the data on using something that kills the cancer daughter cell or the stem cell or the queen is looking after we come up with a drug that all of a sudden kills the cancer and we get rid of it. That's not the concept that I have about cancer. The concept that I have about cancer is that cancer is not your enemy. The concept that I have is that we do not need to kill it. As a matter of fact, cancer is part of your body. I think you need to educate it. The reason that you had cancer is because you did something wrong, or your parents, just like you saw, did something wrong. So your body were trying to repair that area through stem cell plasticity. So for example, you have a wound, you have a chronic condition, you have diabetes, you have inflammation, and then bone marrow sends the stem cells to repair that area. So what happens? That stem cells produces new cells because it has plasticity, right? Now what you need to do, because it gets out of order, it produces cancer in that area. It continues to produce new cells. What you need to do is that, first of all, you need to prevent it so you don't have inflammation, so you don't need to start sending signals to stimulate the stem cells. Second, you need to educate those stem cells, instead of killing them, to become normal. And that's what I do in my practice. It's called cancer stem cell differentiation therapy. And I don't have time, and I can't go through the details, but if you're interested, I have so much studies to show you that you can actually treat the stem cells. Epigenetic instability in cancer. So now this plasticity, which is a good thing, the stem cells are they can produce any cells, they're undifferentiated, which is a great thing that the stem cell can become any cell and repair that cardiac damage or any kind of damage that you have, now becomes a bad thing in cancer, right? So you have to differentiate them. So the epigenetic plasticity involved in normal development under genetic and environmental control that would provide a selective advantage for species adapting to a fluctuating environment, but also provides a strong selective advantage to tumor development. Mechanism of epithelial to mesenchymal transition is an extreme example which is associated with extreme heterogeneity of the gene expression. Embryonal rest theory of cancer basically is a theory that came up 1867, that basically every cancer is produced by some embryonal called stem cells in that area. Differentiation therapy, retinoic acid is an example of it that induces differentiation and is used for promyelotic leukemia. Concept of novel treatment. Although the genetic abnormalities produce defects in growth and differentiation in cancer stem cells, these cells have not always lost the ability to undergo differentiation through epigenetic changes that bypass the genomic abnormalities, thus creating the basis for differentiation therapy. In other words, remember what I told you about epigenetics. One was an inheritable. The second was it's reversible.